Good morning students, I am Shomajit sir. Today I am here to deliver my lecture on topic regulation of gene expression. This is from the chapter molecular basis of inheritance. So without any delay, let's start with the topic regulation of gene expression, that is operon. So let's start. So let's start with the topic of regulation of gene expression. So we can see under regulation of gene expression, so you can see in eukaryotes gene expression occurs with the following levels. So you can see a gene of DNA, then it's converted into introns, that means converted into H and RNA. It has both exon and introns, and then it's converted into mRNA. Okay, and then the translation part. So first is the transcriptional level, that is formation of primary transcript, and second is the processing level, that is splicing and capping. Third is the transport of mRNA from nucleus to the cytoplasm, and fourth is translational level, that is formation of polypeptide. So you can see the transcriptional level, that is conversion of DNA to mRNA. Okay, so mostly the H and RNA, where H and RNA is converted into an mRNA, that falls under processing level. So this transcription level and processing level combine to form a transcription process. And next then the mRNA is transported from nucleus to cytoplasm. So from nucleus the mRNA comes out in the cytoplasm, and the cytoplasm, the translational level starts, where mRNA is converted into protein. So under regulation of gene expression, the metabolic, okay, so it is the metabolic, physiological and environmental conditions which regulate the gene expression. So example we have in E. coli, the beta galactosidase enzyme hydrolyzes lactase into galactose and glucose. So you can see in E. coli, the beta galactosidase enzyme hydrolyzes. Okay, so it hydrolyzes. So in E. coli, the beta galactosidase enzyme hydrolyzes lactose into galactose and glucose. So in the absence of lactose, the synthesis of beta galactosidase stops. So the development and differentiation of embryo into adults are a result of expression of separate or several set of genes. Okay, so the development and differentiation of embryo into adult as a result of expression of several set of genes. So if a substrate is added to growth medium of bacteria, a set of gene is switched on to metabolize it. It is called induction. So when a metabolite, that is product, is added, the genes to produce it are turned off. This is called repression. So you can see the E. coli growth medium. Next we can see the operant concept. Here each metabolic reaction is controlled by a set of genes. So all the genes regulating a metabolic reaction constitute an operant. So example, lac operant, tryptophan operant, and ARA operant, that arabinose operant, HIS, histone operant, and valine operant. Okay, so there are many operants up there. So you can see in a operant, you can see the regulatory gene and structural genes. Okay, so in the regulatory genes we have promoters, operators, inducer. Okay, but in the structural genes we have like Z, lac Y, and lac A in case of lactose operant or lac operant. So lac operant is an operant controlling lactose metabolism. So it was proposed by Francis Jacob and Jacques Monod. So we'll study that it is proposed by Jacob and Monod. So you can see in a lac operant, you can see the promoter. So in the regulatory gene, you can see the promoter region, operator region, and in the structural genes we have lac Z, lac Y, and lac A. So lac operant consists of a regulatory or inhibitory gene. So it codes for repressor protein. Next we have three structural genes, that is Z gene, Y gene, and A gene, that is lac Z, lac Y, and lac A. and next we have an operator region. So genes in the operator functions together in the same or related metabolic pathway. So lack of operon, you can see a regulator gene and structural genes. Now you can see the functions of Z gene, Y gene and A gene. So Z gene, it codes for beta galactosidase, that is it hydrolyzes lactose to galactose and glucose. Then we have Y gene, that is it codes for permease, that means it increases permeability of the cell to beta galactosides, that is lactose. And A gene, it codes for transacetylase. So one is beta galactosidase, permease and transacetylase. Okay, so next you see the lactose operon or lac operon, that is in absence of an inducer. So you can see an operon where a regulator gene, okay, you can see here the regulator gene is forming mRNA and then it forming repressor protein. So you can see the structural gene is here switched off. Okay, so if there is no lactose, that is inducer, so lac operon remains switched off. The regulator gene synthesizes mRNA to produce the repressor protein. This protein binds to the operator region and blocks the RNA polymerase movement. So the structural genes are not expressed. So this is the role of lac operon when the inducer is absent. Okay, so you can see the structural gene, it doesn't have any function. Gene ZYA, it will not produce beta galactosidase, transacetylase and permease because the entire gene is switched off. Now the next option or next type is in the presence of inducer. So same gene in presence of inducer, what happens? So you can see here in presence of enzyme, you can see the gene Z, Y and A is switched on mode, and here transcription and translation is done. So once it does transcription, then mRNA is produced, and then mRNA is converted into protein. 3 different proteins are produced. You can see beta galactosidase, permease and transacetylase. These all help in lactose metabolism. So you can see here inducer is forming mRNA and then repressor protein. Then you can see and one inducer is the lactose. Okay, so if lactose is provided in growth medium, the lactose is transported into E. coli cells by action of permease. So lactose inducer bind with repressor protein. So repressor protein cannot bind to operator gene. The operator region becomes free and induces the RNA polymerase to bind with the genes. Okay, and then the transcription starts. So you can see here then the glucose or galactose cannot act as inducer for lac operon, and regulator or regulation of lac operon is repressed by, okay, so regulation of lac operon is by repressor called negative regulation. So what is negative regulation? Regulation of lac operon by repressor is called negative regulation, and lac operon is under control of positive regulation as well. So this was all about the lac operon when there is a presence of inducer and when there is a absence of inducer. Thank you, student. This was all of the entire lecture. We have discussed about the topic operon, that is regulation of gene expression. So thank you, students. Next meet in the next class.